Up next, unlocking the secrets of human memory by chasing bats in the Israeli countryside. Sounds like a strange way of doing research, but it is showing promise. But first, an intriguing story from Israel, where one researcher is starting to decode the mystery of human memory by tracking the movements of bats. The ceiling of this cave is crawling with bats, Egyptian fruit bats. If you want to catch some for brain research, this is the perfect place to stretch a net.
Bats are mammals, which means they are very similar to us in terms of their brain structure. And I'm studying learning and memory in general, the neural basis of learning and memory, and in particular a brain region called the hippocampus. The hippocampus is a brain region that is crucial for spatial memory and episodic memory, that is memory of what happened when and where. This is what we normally call in everyday language memory. I remember that I visited Paris a year ago. If those regions are damaged bilaterally, meaning on both sides of the brain, then you cannot remember absolutely anything new. 99.9% of what we know about the hippocampus comes from rats, rat studies. And I, a few years ago, decided that it's really a problem if everything we know about this important brain region is just from one species, the rat.
Tonight, Nahum and his partner Asaf are looking for heavyweights.
So this is the bat that is too small for our needs. He weighs 120-something grams.
He has let go. Those that weigh at least 130 grams get a haircut. This is where they will carry a special load. Will be attached with glue that will eventually dissolve.
Laboratory rats are only living in the laboratory. They don't live in the wild by definition. A laboratory rat will not survive in the wild. We know a lot about navigation and spatial memory in rats in the laboratory. We don't know anything about spatial memory or representation of space in the brain in the wild.
So these bats become the first ever to be outfitted with GPS weighing just 7 grams.
And I think it makes a lot of sense not to just look at a scale of a few meters in the laboratory, but also to scale up to geographical scales, tens of kilometers.
After a restorative drink, the experiment goes airborne. 2 weeks later, the research team gets a signal from another cave. As expected, the glue is let go and the GPS has fallen off. They need to find it. On board is a flash card that's recorded the bat's activity. Here is what it looks like. A trip night after night to the same feeding tree.
There is a very famous theory that says animals in general have cognitive maps, and that the map is stored literally within the hippocampus. We really want to at least understand at the behavioral level, are they using strategies in the wild that are thought to be hippocampus dependent or not?
They're analyzing the data now and hope to report soon. Meanwhile, Israel's Batman has 2 other experiments on the go. Both use fruit bats that are caught in the wild and kept in his lab. And both use probes that are inserted into the brain. This is a crawl test. You're looking down on a bat that's moving around a small space. This replicates a standard test given to rats. They are hearing and seeing the activity of neurons in the hippocampus, called place cells.
This neuron will be active, let's say, only when the animal is in this location. And another neuron, another place cell, will be active only when the animal is in this environment. And a third neuron will be active only when the animal is in that location, the environment, right? So you get blobs of activity for different neurons, and together they form kind of a map-like memory of space. And what I found is, first, that there were place cells in the bat hippocampus that were very similar to rats. But on the other hand, I found something very different.
There was a difference in the brain's biggest wave, the theta oscillation.
Just by using a different species, I can have another angle at this question of what is the function of the hippocampus.
The second experiment begins with training. It's done in the lab's flight room.
We're training them to fly and touch 3 balls in a sequence and stay in the air at least 1 minute before they go back to their cage to get a food reward. Food reward is just a piece of banana. These are fruit bats, and they'll do anything for a banana.
They want the bats to map these surroundings. They'll see how they map them by looking inside their brains. To do that, he's building a special portable brain probe. He's using wire that's much thinner than a human hair to make electrodes. Once prepared, 4 of them go on a device called a microdrive. It's surgically implanted on the bat's head, and the electrodes are lowered into the hippocampus. The rest of the gear picks up, amplifies, and transmits the signal. Early trials show the bats can easily carry it in a backpack, and that it does send a good signal. The design of the flight room makes sure of that.
It's enclosed in a shielding, an electrical shielding called also a Faraday cage, that allows to block off completely signals from the outside. We also have special acoustic foam on the inside of the walls. It will reduce the reverberations, the echoes.
The room also allows them to study bats in varying degrees of light and dark.
In complete darkness, they use sonars, those brief emissions of sound, and they allow them by the echoes to reconstruct a three-dimensional image of the world based on sound alone. And I can record exactly when did the animal emit the calls. I can precisely, on the millisecond scale, know when the sensory information from the environment got to the bat's ears or to the animal's ears. And actually, I was able to show that the timing of those calls, the sonar calls, has great influence on the activity of those place neurons. And this is something you cannot do in a rat, because the rat is running around, it's sniffing, has a smell, it's touching. It's very difficult to get hold on what is it exactly sensing, what sensory information is impinging on its senses. This is another advantage of using a bat over a rat for studying the hippocampus.
The hippocampus is affected in diseases like Alzheimer's. To solve that kind of dysfunction, science first needs to understand function. The bat is giving us important new information.